Good afternoon, everyone. I'm Bhargavi, and I'm a PhD student working with Vineet Bafna at UC San Diego. Today, I'll be presenting our work on AmpliconSuite, which is a complete workflow for analyzing focal amplifications, and in particular, those that involve ecDNAs. Focal amplifications are regions of the genome that are duplicated multiple times, which leads to an increase in number of copies of specific genes. These amplifications often drive cancer by overexpressing oncogenes. On the left, we see a fish image of a focal amplification of EGFR, and in the bottom, we see EGFR, but this time amplified through extrachromosomal DNA or ecDNA, which are segments of DNA that are not incorporated into chromosomes but exist independently within the cell. We know that ecDNAs are known to amplify oncogenes to very high levels and contribute to rapid tumor growth and resistance to therapy. On the right, we see that ecDNA is prevalent across many tumor types, and it's also associated with worse patient survival when compared to other amplifications. These focal amplifications often come with complex structures, and so they present unique challenges to their analyses. AmpliconSuite tries to address these challenges by packaging tools into one comprehensive workflow for their analysis as well as identification. So let's start with an overview of the AmpliconSuite pipeline. We start with paired in whole genome sequencing data, which can be in FASTQ or BAM format. Um, then we use existing tools like BWA or SAM tools to um, align the reads, followed by seed detection using CNV kit. This identifies candidate regions for focal amplifications by looking at high copy number regions. And then we filter some of the seeds to remove non-local, non-focal amplifications and repetitive sequences. And then structure analysis is performed using Amplicon Architect, which is a tool that constructs local genome graph of amplified segments and structural variants while incorporating the copy number information of these regions. So the graphs it constructs remains consistent with the copy number profile of the entire amplicon. Finally, Amplicon classifier uses all of the information thus far to categorize these amplifications into distinct types, such as those that are extrachromosomal DNA, those that look like breakage fusion bridge, those that are linear, and so on. And then there are also workflows for understanding the origin or formation mechanism of a given ecDNA that are also integrated into the same pipeline. For example, here on the top, we see an ecDNA that looks relatively simple, which is consistent with an excision-like formation mechanism where the two ends of a DNA segment get cut off and later circularize. And in the bottom, we see a much more highly rearranged structure, which is more consistent with a chromotipsis-like formation of this ecDNA. There are also integrations to platforms like GenePattern and Nextflow, which facilitates usage without needing to write code. And then finally, AmpliconRepository.org is the website where researchers can upload, share, and build on other people's findings, and also make it publicly available if they choose to. I would like to first mention that this work, the primary authors of this, the first authors of this work are Jens Lubeck and Edwin Hong at UC San Diego. And I'd like to say thanks to Vineet Bafna and all the other collaborators on this project, and special thanks to all of the users of AmpliconSuite who provided valuable feedback and testing.